We've taken basically a technique that we use for nuclear medicine, which we use to radiolabel compounds and see where they go in the body, and applied it to this compound that's found in the environment called perfluoroalkylated substances. And so by using this technique, we can make this essentially a tracer of that compound and see where it goes in the body, how long it stays there, and the different clearance pathways of the different chemicals. This paper is interesting because it really shows how we can take techniques from nuclear medicine and from our imaging studies and really apply them to more environmental compounds. You know, when people think of radiochemistry, you know, typically they thought of tritium or carbon-14 or these very long-lived compounds, you know, and doing these pharmacokinetic studies. And now we have a whole host of different radioisotopes with different chemical properties, and we really have these nice tools that we can use for different applications, let's say outside of nuclear medicine, like environmental cleanup applications. PFAS have been found in many different environmental sites, so they're leached out of the groundwater and they accumulate in the human body and in the plants and animals. And so because they're accumulating, people are very concerned about the environmental and health implications of these compounds. They're found a lot in groundwater. So let's say that the PFAS are found in fast food wrappers or different kinds of material, that sort of thing. So those materials, they end up in the landfill, and then when it rains, the water leaches them and they end up in the groundwater, which ends up in your drinking water. And because they're very persistent, which means they don't break down in the environment, means that they stay in the water for a very, very long time. So there's a lot of initiatives to try and figure out how to clean up these compounds. Using this new technique, we can really screen compounds very rapidly. So imagine if we can label many different classes of these perfluoroalkylated substances and see the differences in the chemistry and where they go in the body. Let's say we can find different compounds that may be excreted faster, may actually be not as persistent in the body, and we could tell those differences quite rapidly with this radio tracer technique. Conventionally, tracing these compounds is very difficult. These compounds are not UV active and they're actually very, very difficult to detect. And so there are some techniques where you can detect total fluorine concentration, but that doesn't give you an idea of which compound the fluorine is attached to. With our method, we can actually tag the intact compound with a fluorine-18 radio tracer, and it gives us sort of a handle so we can see where that compound is going, and we can make very, very sensitive measurements. And these sensitive measurements are actually probably the most important thing because it's so difficult to detect in other methods. It's a much quicker and less time-consuming method to look at where these go. So the drawback is that we can only measure for short periods of time because the radioactive tracer is really short. So it's actually a very complementary method to these more, in-depth, complicated methods to do that. When people think of radiochemistry, most of the time they think of, carbon-14, tritium, these long-lived radio tracers, which really present a disposal problem at the end of the day. With these short-lived radio tracers, we can do rapid screening, we can develop quick chemistry, and look at these compounds very, very rapidly, and really look at the chemistry of a wide variety of compounds.